Thank you, Brenda, for your kind introduction. Hello, everyone. My name is Govindaraj Kumaran. I'm a CMC reviewer in OPQ, Office of Pharmaceutical Quality, CDER FDA. So I'm happy to be here to present nitrosamine risk assessment in type 2 DMFs supporting GDUFA applications. A quick disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. I would like to start off with OPQ's overall goal and mission. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. This slide provides the learning objectives of my presentation. First, I will discuss the formation of nitrosamines, and then nitrosamines in drug products and FDA guidances, potential root causes of nitrosamines in API, nitrosamine risk factors in API, recommendations to API manufacturers, approaches to determining acceptable intake limits, nitrosamine control strategies, and finally, summary. So the nitrosamine is a class of compounds in which the nitroso group is attached to an amine. One of the most common way to form the nitrosamine is the nitration of secondary amine under acidic condition. If a nitrosonium species is generated from the nitrosating agent, then the secondary amine will readily react with the nitrosonium species, and it will produce the nitrosamine. Tertiary amines may undergo dealkylation to form secondary amine, which may undergo nitration to form the nitrosamines. And amide solvents may decompose to secondary amine, which may undergo nitration to form the nitrosamines. So the nitrosamine is a probable human carcinogens. They are potent genotoxic reagents, and the nitrosamines are referred as a cohort of concern in ICH M7 guidance. Because of their high carcinogenic potency, they are regulated more tightly than typical mutagenic impurities. The TTC limit, that is a threshold toxicology concern limit, should not be applied to the nitrosamines. This slide describes the small molecule nitrosamines in drug product. The first nitrosamine issue was reported in June 2018. So in June 2018, FDA informed the presence of NDMA, that is N-nitrosodimethylamine impurity from a valsartan manufacturer, and then later small molecule nitrosamines in drug product were reported, and some of them are listed here. Then to address these small molecule nitrosamines, FDA first published the nitrosamine guidance in September 2020, and then it was later revised in February 2021. The guidance is control of nitrosamine impurities in human drugs. So this guidance actually provides some basic information about the nitrosamines, and also it provides a control strategy for the nitrosamines in drug substance and drug product. More importantly, this guidance recommended control limits for certain small molecule nitrosamines. Some of the small molecule nitrosamines are listed in this table. At the time, these small molecule nitrosamines are probable nitrosamine impurities in the drug substance and the drug product. When the small molecule nitrosamine impurities were reported in the drug products, simultaneously NDSRIs in drug products were also reported. NDSRIs are nitrosamine drug substance related impurities. They are class of nitrosamines sharing structural similarity to the API. FDA identified this issue in a public statement on November 18, 2021. So the presence of NDSRIs in drug substance and drug product added additional complexity to this nitrosamine issue, primarily because determining the acceptable intake limit for these NDSRI is more challenging than the small molecule nitrosamine impurities, primarily because these the NDSRIs are unique to each API, and there is no safety data or carcinogenicity data reported for these NDSRIs. So determining the acceptable intake limit for these NDSRIs is more challenging. So that becomes a huge issue in the pharmaceutical industries. To address this issue, FDA again published one more guidance in August 2023. That is recommended acceptable intake limits for NDSRI, that is rail guidance. So the major component of the rail guidance is the CPCA, carcinogenic potency categorization approach. Using CPCA, the rail guidance categorizes the NDSRI into 5 different categories, and the associated acceptable intake limit ranges from 26.5 nanogram per day to 1,500 nanogram per day. You can find this guidance in this web page, and the link is provided here. And this web page also provides some additional information on acceptable intake limits. There are 2 tables are there in this web page. Table 1 contains searchable list of 264 hypothetical NDSRIs, potency categories, and associated recommended acceptable intake limit. And the table 2 contains list of NDSRI for which FDA has identified acceptable intake limits based on compound-specific data or read-across analysis. This web page will be updated periodically. So moving on to the next section, root cause of nitrosamines in API. The root cause of nitrosamine in API can be categorized into 3 categories. First is intrinsic properties of API, process-related, quality of raw materials. Intrinsic properties of API. Suppose if an API has a nitro group and a secondary or tertiary amine in the same molecule, then there is a possibility that the API can generate the nitrosamine impurity. For example, in the case of the ranitidine, this ranitidine has nitro group and a tertiary amine in the molecule, and it was reported that the ranitidine can generate the NDMA impurities by the intermolecular degradation of API without the requirement of any exogenous nitrate source. So the other category for the root cause of nitrosamines in API is process-related. In the most common pathway to formation of nitrosamines, 3 factors are required: presence of nitrosamines, presence of nitrosating agent, and conditions conducive to nitrosamine formation. Presence of nitrosamine, nitrosable amines. Excuse me, let me have some water. Okay, presence of nitrosable amines. Amine functional groups are very common on API molecules, isn't it? Almost 20% of the approved drug products contains the amine functional groups. And the tertiary and secondary amines are common bases used in the synthesis. And amide solvents are very common in the API manufacturing process. They can significantly degrade under high temperature to form secondary amine. For example, dimethyl formamide. Dimethyl formamide can degrade at high temperature to form the dimethylamine, and that amine could cause the formation of the nitrosamine impurities. So the other factor for the formation of nitrosamine is the nitrosating agent. So the nitrosating agent can be intentionally added as a nitrate salt or nitrous acid, or It can be an impurity from another reagent, such as nitric acid or sodium azide. The less common nitrosating agent is alkyl nitrates. Alkyl nitrates may be intentionally added to the API manufacturing process, or it may be an impurity from a rearrangement of nitroalkanes. So the third factor is the conditions conducive to nitrosamine formation. Acidic condition facilitates the nitrosating reaction. So the other process-related root cause of nitrosamine is the oxidation of 1,1-disubstituted itrazine. In the case of 1,1-disubstituted itrazine, you don't really need any external nitrosating agent. Even the molecular oxygen can oxidize the 1,1-disubstituted itrazine, and that will generate the nitrosamine impurities in the API manufacturing process. Nitrate formation from oxidation of hydroxylamine is known, or nitrate release from nitroaromatic precursors is also known. And the other root cause of process-related root cause of nitrosamine in API is fluid bed drying. Nitrogen oxides in the intake air of the fluid bed drying can generate the nitrosamine in the API. And use of activated charcoal also can pose the nitrosamine risk, because when you use the activated charcoal in the final stage, especially in the final stage, and if there is a secondary amine present in the process, then there is a risk for the formation of the nitrosamines. So the other process-related root cause of nitrosamines is a poor operation design of the process. Suppose if nitrosamine is formed in the API manufacturing process, and because of the poor operations or design of the process, the adequate purging may not happen for the nitrosamine impurities. Then that impurity will carry over to the final API product. And cross-contamination of nitrosamine impurities in multipurpose facilities has been reported. So the other category in the root cause of nitrosamines in API is quality of raw materials. Use of raw materials containing impurities. So the amine and nitrosamine impurities were found in the fresh solvents. Some of them are listed here. And the use of disinfected water can generate the nitrosamine impurities. And nitrate impurities are found in the several reagents, such as sodium azide water and sodium carbonate, sodium bicarbonate, and sodium hydroxides. And it has the recycled materials, such as solvents, reagents, and catalysts, are confirmed source of nitrosamines. So these are all some of the root cause of nitrosamines in API manufacturing process. So moving on to the next section, small molecule nitrosamine risk factors in API. One of the risk factors for the formation of small molecule nitrosamine is the use of secondary amine or tertiary amine bases or tertiary amide solvents, and the nitrosating agents are deliberately added to the process, or if the nitrosating agent is present as a significant impurity in the other materials, especially if these precursors are used in the same or adjacent steps. The other risk factor for the formation of small molecule nitrosamines in API is presence of 1,1-disubstituted itrazines or their imines in the process. Because I said that 1,1-disubstituted itrazine can react with the molecular oxygen, and it will produce the nitrosamine impurity. How about the NDSRI risk factors in API? If the starting material, intermediate, related substances, or API have a secondary or tertiary amine functional group, and nitrosating agents are deliberately added to the process, are present as a significant impurity in other materials, especially if the precursors are in the same or adjacent steps. Suppose if the process used the fluid bed drying or jet milling process, and the API or counterion contains secondary amine, then there is a risk for NDSRI in API. If the activated carbon is used in the purification of the crude material, crude or final API, and the API contains, API or counterion contains secondary amine, then there is a risk of NDSRI in API. Recommendations to API manufacturers. Look at the API synthetic route holistically. Don't look at piece by piece when you do the risk assessment of the nitrosamines. Audit the starting materials, intermediate vendors. Understand the risk coming from the purchase materials. And if a risk of nitrosamine is identified, confirmatory test of batches should be conducted using sensitive and appropriately validated methods. If nitrosamines are detected, then perform a root cause analysis and implement changes to manufacturing process to prevent or reduce nitrosamines, add control wherever it is necessary, and report the implemented changes to FDA. Approaches for determining acceptable intake limit of NDSRI. To determine the acceptable intake limit, first you check the AI limit appears on the table 1 or table 2. You can find the the link for the I provided the link for table 1 and table 2. You can check that table and then first check those tables to see whether that particular NDSRI is listed in the table 1 or table 2. If AI limit cannot be found in the above tables, determine using CPCA to determine the acceptable intake limit using CPCA recommended in the rail guidance. If they are unable to meet their CPCA-based acceptable intake limit, they can use read across analysis from a surrogate to determine the acceptable intake limit, or they can submit the compound-specific toxicology data to determine the acceptable intake limit. Certain nitroso compounds, for example, N-nitrosamide, N-nitrosoguanidine, and N-nitrosourea, the CPCA, these nitroso compounds are out of scope of CPCA. For these type of N-nitroso compound, alternate approaches such as read across analysis from a surrogate candidate or compound-specific toxicology data may be used to determine the acceptable intake limit. Nitrosamine control strategies. The ICH M7 provides 4 options to control the mutagenic impurities. The API manufacturers can use that 4 options as a control strategies, but it has to be the control strategies should be supported by the appropriate data and justification. Suppose if multiple nitrosamines are controlled in the specification and the total level of nitrosamines exceeds the acceptable intake limit of the most potent nitrosamine based on the MDT, the manufacturer should contact the agency. I provided the contact information in the final slide. You can contact the agency in case if the the total level of the nitrosamine exceeds the level of most potent nitrosamines based on MDT. Challenge question 1. Fluid bed drying or jet milling in the API manufacturing process has potential to form NDSRI when API contains primary amine, secondary amine, tertiary amine, or amide. The answer is secondary amine. Challenge question 2. Which of the following nitroso compound is out of scope of CPCA? N-nitrosamines, N-nitrosoamides, N-nitrosoguanidines, N-nitrosourea. all the above except A. The answer is all the above except A. In summary, nitrosamine formation may be a risk in API. Process understanding is essential to mitigate the risk. Build quality into the process to minimize or eliminate the nitrosamine formation. Always take a holistic approach when assessing the nitrosamine risk. If you have any questions related to nitrosamines in API, use CDR-OPQ-inquiries at fda.hhs.gov. We will get back to you with the answer. And with that, I thank everyone. If you have any questions, I'm happy to answer in the question and answer session. So I would like to invite the next speaker, David, to the podium. Thank you.